Okay, thanks for everyone who's joined so far, and hopefully we will get some more coming in. It's my honor to introduce Cleo Valentine. She is a senior design researcher in neuroarchitecture at HKS, which I believe is an architectural firm, and vice chancellor's doctoral scholar at University of Cambridge, specializing in architectural neurophysiology and bioethics. Her work explores the impact of architectural forms on neuroimmunological activity, with a particular focus on the relationship between neuroinflammation and the built environment. The aim being to uncover new insights that can inform architectural practices to promote better health outcomes. She currently serves as senior research and innovation lead at the, I guess, fairly new International Center of Neuroarchitecture and Neurodesign, which is a collaboration between UCL and the Research Institutes of Sweden, and is also the neurophysiology lead at Beautiful Places AI. Her research partnerships include collaborations with Heatherwick Studios' Humanize campaign, and she also maintains affiliations with University College London Spatial Cognition Lab, as well as MIT Department of Urban Studies and Planning, and the University of Cambridge Design Engineering Center. So a lot going on. And without further ado, I'll hand it over to Cleo to walk us through everything.
Right on. Thank you so much. Okay, I'm going to share my screen here. Let's see how we crack on. Okay, cool. What can you guys see right now? I want that. How's that looking?
I still don't see anything on my side. So
Yeah, still just you.
Just
sometimes you need
okay.
there's another click
Oh, you know what
sometimes.
it is?
There we go.
There's a share
There,
button.
there
Yeah,
we
Ah,
go.
well, there
you
you
need
go.
to actually press the share button, it turns out.
Yeah.
Okay, cool. Let me see here. Okay, perfect. Onwards we go. So you can see my main screen. Whoop.
Yep, looks
Sorted.
great.
Okay, wonderful. Well, first of all, thanks so much for having me and for that very generous introduction. I'll just crack on into it. But I'm Cleo Valentine, so I'm going to speak to you a little bit about the field of architectural neuroimmunology, which is really the term that I use to describe the relationship between the built environment, the immune system, and the brain. Which screen are you guys seeing right now? Are you seeing my primary one? Because I've got three different ones, and I'm never totally sure.
We're seeing architectural neuroimmunology, and then we're seeing this nice animation of the brain slices
Sorted.
or something, I guess. Yeah.
Stunning. Okay, cool. Well, then let's hop in. So neuroimmunology is actually a relatively new discipline in and of itself, and it really examines this very intimate interplay between the nervous system and the immune system. And both of these systems play a really critical role in mediating the body's interaction with its environment. And this very much includes the built environment. But specifically, the work that I'm going to take you through today, it really explores how the built environment can elicit something called neurophysiological stress responses, and what that means for both public health and also kind of the built environment profession more generally, and how we can really engage with this research going forwards. So for anybody who's ever seen one of my lectures, this may be a quick recap, but I always really like to start with the review of some of the physiological processes that I'm going to be talking about today. So this is a paper or diagram from a recent paper of mine, and all of these have QR codes in the bottom. But in this diagram, what you can see is a really simplified map of a stress event. So stress in this instance, it refers to it's any real or perceived threat to one's psychological or their physiological well-being. So this means that a stressor can be something like a physically traumatic accident, like a car accident, or actually something emotionally distressing or spatially disorienting. So the term stress here is used in a very, very broad way. But in the specific model that we use, acute or short-term stress triggers something called allostatic processes in the body. So this is more commonly referred to as the body's fight or flight response. And the response, it really aims to restore the body to a state of balance or equilibrium, which is also known as homeostasis. And so it does this through eliciting specific neurophysiological changes that actually help us to navigate or evade the stressor. So for example, you'll see an elevated heart rate to pump more blood to the muscles and the hearts and the vital organs. You'll see an increased supply of oxygen in the brain to heighten mental alertness. And although these kind of stress responses are actually incredibly effective in acute short-term events, they're actually protective and necessary for survival. Chronic stressors can actually lead to prolonged neuroendocrine responses in the body that have the potential to really compromise the immune system and cause long-term damage to the organs and tissues. And this very much includes the brain, which is what I primarily look at. This is a slight aside. I'm not going to get into this paper, but if people are so inclined later, it's brand new that came out of our research group. And it actually highlights the extent of the possible impact of this architecturally mediated allostatic overload response that I'm talking about. But it specifically looks at neuroplasticity and neurogenesis. So the paper really proposes a theoretical framework which suggests that chronic exposure to stress-inducing architectural environments can actually alter brain functions in key regions, specifically the hippocampus and the prefrontal cortex, which can actually contribute potentially to profoundly adverse mental health outcomes. So that's a brand new paper that just came out of our research group, so I'll just put that in there, but we won't get into the details today. So on this field more generally, what we're increasingly seeing here is compelling evidence supporting the effect of architecture and the built environment on this stress response that I'm talking about. But to date, there's been not a tremendous amount of consideration actually given to understanding the extended physiological outcomes associated with these responses. And so, but what I mean by this is, what are the downstream effects of this architecturally mediated stress response? And although there's not a ton of research in architectural disciplines, there actually may be emerging evidence from adjacent fields that can shed some light on this. So specifically, the emerging field of neuroimmunology that I mentioned may be able to do so. So as I mentioned very briefly, neuroimmunology is a relatively new discipline. It's really kind of come into itself in about the last 10, 20 years. And it really examines this interplay between the nervous system and the immune system. And actually, one kind of seemingly simple, but actually incredibly profound finding from neuroimmunological research more generally is that these physiological stress responses, they're not discrete singular events. Instead, these stress responses, they actually can set off an inflammatory cascade that moves through the entire body and kind of elicits specific changes within various pathways. And that's kind of what we're looking at here. So one known outcome of chronic or extended exposure to these stress responses is neuroinflammation, which is my kind of primary topic of interest. And neuroinflammation quite simply refers to inflammation of the brain. And chronic neuroinflammation is associated with the development of a number of conditions, so ranging from depression, but also Alzheimer's and ALS. And I focus on neuroinflammation as opposed to other forms of systemic inflammation, in part actually because the presence of these conditions is increasing exponentially. And this is particularly the case in urban areas. So just to kind of back up and frame this a little bit, a lot of the emerging evidence in neuroimmunological stress research suggests that the concept of a threat or a stress response is actually understood in a really contextual way. And so what the research suggests is that environmental features, including features within the built environment, such as window size, ceiling height, lighting arrangement, or the presence of specific visual patterns, which inherently are actually non-threatening. They don't actually pose any direct risk, but they can also trigger these neurophysiological stress responses. And one way that these negative responses to our environment and the built environment have actually been indicated is by early activity in the visual system within the brain. And so when I say early activity, I'm talking about kind of seconds of exposure. But what hasn't really been considered yet is the impact, as I mentioned, of this extended visual exposure to these architectural forms. So what happens when you are exposed to these environments for minutes, or for hours, or for days, or for months, or for years? And so this has very much been the focus of the first piece of research that I'm going to be showing you right now. I always have to put a health warning here. Some of the images that I do display actually they contain visual patterns that can actually induce visual stress. So if you are sensitive to visual patterns, or you do have conditions like epilepsy, I would advise maybe looking away for the next few slides. So one of the features that we've been looking at are something called high contrast, regular repetitive visual patterns. But kind of what do I really mean by that? This actually builds on the work of Arnold Wilkins and Ian Hosking over in the UK. And the research we've been working on examines the impact of these regular, repetitive, high contrast patterns, so actually just stripes, on the human visual system within the built environment. And interestingly, exposure to these patterns, it can actually overstimulate the primary visual cortex. And this is especially the case when the patterns exhibit a spatial frequency of around 3 cycles per degree of the visual angle. And I mean, probably don't need to explain this to you guys, but spatial frequency refers to the number of changes in visual information, such as light or dark patterns, per unit of space in an image. So this is a very specific manifestation of geometric principles, really. And what I mean by this kind of overstimulation of the primary visual cortex is that what we're seeing is an increased metabolic response in the visual cortex. So that means it's working very, very hard to process the visual scene. And as a result, it's using a lot of oxygen. And this overstimulation of the primary visual cortex can actually lead to visual discomfort, impaired task performance like reading rates, and in slightly more extreme cases, it can trigger migraines and seizures in susceptible individuals. It can also cause disorientation and visual distortions. So we're seeing a broad range of symptoms resulting from visual exposure to specific geometric patterns. While the presence of these patterns, they may not seem particularly consequential, once you notice them, they actually become really difficult to ignore. So I've spoken to several people who actually report being unable to enter specific environments because the visual patterns trigger headaches and migraines and visual distortions. Also, when I've showed this research to architecture and engineering firms, I've actually had designers tell me that clients have complained about specific designs impacting people so much that they've had to retroactively alter the space after the project completion. So one specific example was a library where students were complained that the acoustic panels were triggering migraines. And I recently had a similar experience where the acoustic panels in a lecture theater were eliciting strong visual distortions. So it actually is incredibly pertinent to kind of the design discourse right now, I think. I do always, academic background, it's very important that I highlight that people have varying degrees of visual sensitivity. So approximately 10 to 20% of the population, based on our current knowledge, may not experience any significant impact when they're exposed to these visual patterns. But our current research indicates that around 10 to 15% might develop clinically significant responses. And this is a subgroup that we're currently evaluating. Though I will say I've got a new paper that we're just working on right now, and that 10 to 15% is looking a little bit higher. So kind of watch this space, but there is a variation in the ways in which people respond to these environments. So I always like to kind of back up a little bit and try and really understand why these patterns might cause discomfort. So one theory really suggests that our visual system evolved to efficiently process images commonly found in natural environments. So visual processing is most effective actually when images have spatial characteristics found in organic material. So natural images, although you can see here, they're very diverse in their content, they actually share statistical features, specifically this 1/f Fourier amplitude spectrum, which actually facilitates efficient encoding by the cortical neurons, which is really, really interesting. But, departures from these natural image properties, many of which we're seeing more commonly in contemporary urban environments, have the potential to induce visual discomfort. And so, for lack of a better term, these unnatural image properties, which can cause visual discomfort, include things like high contrast visual patterns, which we just talked about, strong luminance contrasts, and also high degrees of light flicker, which is something that we're looking at increasingly. And the kind of cortical excitation and the associated visual stress from these sort of unnatural image properties is actually thought to occur because humans lack the ability neurologically to process spatial features which are rarely found in natural organic environments. So that's a kind of really strong theoretical underpinning behind a lot of this research. This is a slide aside. I put this in every single lecture I do. The quality of the video is not very good, but I think it illustrates the point so well that I always like to include it. I'll play it in 1 second, but I'd like to give a little bit of context because, although evidence suggests that, humans have been on Earth for roughly 200,000 years or so, there's actually only evidence of urban settlements dating back about 6,000 years. So this really suggests that as a species, we're actually quite new to urban living. So just to give you a sense as to the rate of urban change, I'm going to play a video in a second from the New York Times, which shows a time-lapse animation covering about 500 years of urban development on Manhattan. So I'll just let that play for a second.
We're not hearing audio. I
Yeah,
don't know
we're
if you...
just taking it in. There's no
All
audio.
right,
It's
perfect.
just
If there isn't, then that's great. I'm enjoying the rides.
it's
Just
it's
thought I'd
it's
mention
it's an
it.
intentional pause.
Okay,
But
perfect.
yeah, so this is a video that I love. I always include it. It's really just a time-lapse animation. And what I think it really highlights is that urban environments change at an incredible rate. However, our biology, a little bit slower to adapt. So one figure suggests that it takes about 4,000 lasting generations for an evolutionary trait to become fully embedded within a species. And so what this suggests is that our environment changes at a rate that our physiology may really struggle to keep pace with. So to kind of summarize the theoretical foundations here, acute exposure to these regular, repetitive, high contrast patterns in architecture has been linked to cortical excitability and visual discomfort. But the impact of extended exposure, so by that I mean kind of minutes or hours, to these patterns in architectural design, their impact on neurophysiological stress response has not actually been directly studied. So this is very much what we're looking at in our paper that actually came out quite recently. So there are a few different ways that we can examine the impact of these patterns on neurophysiology. The first is mathematical analysis. So we can evaluate the spatial properties using a Fourier analysis, and we can assess the luminance contrast or the chromaticity in the architectural image. The second is that we can do user impact assessment. Visual discomfort's actually reasonably well self-reported. So we can also measure task performance and preferences using Likert scales and self-reported visual discomfort metrics. And the last thing is that we can use neuroimaging. So we can record cortical responses using techniques like fMRI and FNIRS, functional near-infrared spectroscopy, which I'll explain in a little bit. As I mentioned, not all high contrast patterns will induce visual stress. Factors such as the pattern size, the spatial frequency, the luminance contrast, the viewing distance, the viewing angle, all will play a significant role in the ways in which these patterns impact the brain and the body. And this is part of what makes architecture so unbelievably difficult to study, is that it's such a dynamically experienced stimuli. But in order to try and engage with some of these questions, we actually designed a two-part study that uses these three methodologies to examine the impact of visual patterns in architectural facades on both the visual cortex and also the downstream stress responses within the body. And the hypothesis really is that architectural facades with highly regular repetitive patterns will result in inefficient cortical processing and increase the level of physiological stress in the body. So in this paper, which very excitingly came out recently, we looked to understand how visual exposure to certain architectural patterns, these specific high contrast, regular repetitive forms, impact physiological stress. And we did this using computational methods and generative AI models. One of the big limitations, I always think that this is kind of an important thing to, engage with, are the limitations that we face in neuroarchitectural research. Because one of the big limitations that we face is ecological validity. So how do we create test environments that mimic the real world while allowing us to manipulate specific spatial characteristics? We need to know which elements of the built environment impact people, but this creates these very kind of inherent trade-offs between realism and experimental control. So I'll just kind of go over these very, very broadly, but there are sort of four approaches that we can use in neuroarchitectural research to engage with these questions. So the first is the use of simple patterns. So one method uses abstract patterns like the pattern glare test, which was that striped circle that I showed you before, which actually, controls spatial frequency and contrast. So it's great for isolating variables with high internal validity, but it lacks ecological validity and doesn't actually resemble real architectural forms. The second is the use of architectural renderings. So renderings offer more realism while retaining control. They improve ecological and face validity, especially when they're photorealistic. But in many instances, although this is kind of changing, the resource intensive show limited views and internal validity drops due to confounding design elements, including things like the 2D nature of a lot of these renderings. Third is photographs. So photos of real buildings increase realism and face validity. So studies show strong correlations actually between photo responses and in-person experiences. Also, I'll add that I'm specifically looking at visual exposure. So when you get into multisensory questions, that changes. But when you're looking at visual responses, actually, they can be quite compelling in some instances. But you can't easily manipulate elements like patterns or contrasts, making kind of causal inference quite difficult, really. And the last is in situ evaluation. So this has the highest degree of realism. So participants engage directly with real environments, but of course it comes at the cost of control. It's hard to isolate variables in dynamic urban environments. Though actually I would say that this approach is actually crucial for validating any in-lab findings. We really do need a combination of all of these in order to paint a very full picture as to what's going on in the built environment. But these are some of the kind of larger scale issues that we struggle with within the field more generally. But we try to engage with this. We tried to engage with these limitations because they highlight the need for new methodological approaches that can bridge the gap between experimental control and ecological validity when looking at the impact of the built environment on the human body and the human brain. So in order to kind of get into this question, we tried to address this by developing a hybrid method that really combines generative AI and a Fourier-based computational or mathematical analysis tool that we're developing in Cambridge called VISTA, the Visual Stress Analysis Tool. And what this approach does is it allows us to generate facade images using controlled text prompts, which enables our kind of systematic variation of visual parameters while allowing us to maintain a consistent base geometry across all of the images. Oh, I forgot I have an animation. And this consistency is actually crucial because it, as I mentioned, allows us to isolate the effects of specific architectural features and kind of trace them back to the neurophysiological responses within the body. So in order to test the effect of a specific facade on neurophysiological responses, we tried to create these controlled stimuli. And this work is actually funded in part by Heatherwick Studios' Humanize campaign. But to do this, we actually use Midjourney, so a generative AI tool, to design these permutations and combinations of a single facade with varying degrees of spatial complexity, geometric form, and visual stress. And next, we applied the computational tool that I mentioned called VISTA, Visual Stress Analysis Tool, that's been developed by our team in the Design Engineering Center at Cambridge. And what this tool does is it allows us to evaluate how architectural patterns affect the primary visual cortex using computational models of the brain. And it does this by doing kind of a mathematical analysis that produces heat maps that allow us to help identify potentially harmful design features and also provide insights into the potential neural inefficiencies that might be caused by these environments. So in the image, you can see here that the regular repetitive wooden feature on the second floor is being identified as a potential stress-inducing visual feature. So these are some of the outputs of the computational analysis, and the yellow area identifies the areas which are potentially most visually stressful or eliciting the strongest response within the primary visual cortex. Interestingly, you can also see the trees lit up. That's actually because it highlights both the features which are closest to organic geometries and the furthest. But this is something we're kind of developing further right now. So what the results revealed were some pretty clear patterns actually in what creates visual stress in architectural facades or elicits this visual stress response. So facades with regularly spaced elements at approximately 3 cycles per degree, as I mentioned. So that's actually about the spacing that hits our visual system's peak sensitivity function. And these exhibited the highest probable stress metrics. And the effect was actually particularly pronounced when these regular patterns were combined with high contrast ratios and consistent repetition across the facade. So specifically, the regularly spaced vertical wooden slats and the vertical metal screening elements produced the most significant indicators of visual stress here. In contrast, the facades with more varied geometric compositions, those that broke up the regular patterns in the high contrast forms, demonstrated substantially lower stress metrics. And I'm happy to kind of discuss these findings and the technicalities in a little bit more detail later. But what's particularly exciting, I think, about this is that our methodology really is trying to offer a scalable, reproducible approach for evaluating visual stress that can be applied quite widely early on in the design process. So rather than designing buildings and hoping that they don't cause discomfort or testing after the fact, we're looking to try and proactively address these health impacts of the built environment during the design process itself. So this was very much a first attempt, and we're really excited about the potential for this. And this is kind of a slight aside, but we're actually in the process of, well, it's under peer review now, a paper that uses VISTA to examine the potential visual stress associated with specific architectural design features across 5 historical periods in Seoul, ranging from kind of traditional Korean architecture from the 1400s to modern skyscrapers. And we're really looking at variables of organic materials, prefabricated construction techniques, and mechanization. And we're really, really excited about this paper, and we'll be presenting at the Seoul Biennale of Architecture and Urbanism in September. So we're really excited about the potential uses of this tool. So this is the second paper, which also uses the same generative AI image set. And in this research, we're really looking to understand how visual exposure to certain architectural patterns, as I mentioned, specifically these high contrast, regular, repetitive patterns, impact the brain and the body more generally here. And so this research is ongoing, and we're actually just finishing the statistical analysis on it now. But in this study, we collected data using something called functional near-infrared spectroscopy and heart rate variability, and we looked to assess the impact of these same visual exposure, or the same patterns of visual exposure in the architectural facades on allostatic activity within the body. So we wanted to look at the brain and also the systemic kind of inflammatory response or stress response, the kind of cascade that happens in the body afterwards. And so in this study, the images of the facades that I showed you before were actually projected at scale in one of our lecture theaters, while clinical biometrics were recorded. And so what I mean by scale here is that we actually projected the facade images on the wall at a 1-to-1 scale to replicate the direct impact on the visual cortex. Just kind of overview, for this, we did 20 participants, heart rate variability and neurological activity, and they were assessed while they're exposed to the projected images of the architectural facades for a total of 25 minutes. So heart rate variability, just for a bit of background, is a key indicator of physiological stress response, and HRV measures the variation in time between heartbeats, with higher HRV kind of counterintuitively indicating a relaxation and state of adaptability, and lower HRV suggesting a state of stress. And FNIRS, this functional near-infrared spectroscopy, it actually uses infrared light to measure oxygenation levels in the brain, which reflect those changes in that metabolic activity. And so this really indicates how much effort a specific brain region, such as the primary visual cortex, is exerting. And so by combining FNIRS and HRV, we're actually able to get insights into both the brain activity and the overall stress response. And the way that I like to describe FNIRS is basically like if an fMRI machine and an EEG had a baby. So it's a cap that actually shines infrared light into the brain, and then that light gets reflected back and we collect it on the scalp. So it's quite dynamic, and it doesn't actually require you to be sitting within a machine, so we can have a little bit more kind of control and agency over the experience of the space. So, we combine that functional near-infrared spectroscopy over the visual cortex with the heart rate variability to try and assess this localized brain activation and that broader stress response. And the experimental protocol basically involved baseline measurements followed by viewing these high stress and low stress facades for a total of 5 minutes for the HRV reading, plus an additional 8 facades that were exposed for 20 seconds each using the FNIRS. And we actually use the neutral gray screen in between them as a washout period. So as I mentioned, we're still processing the data, but so far the findings suggest that the presence of these high contrast, regular, repetitive forms do actually elicit the systemic neurophysiological response. And we're really hoping to have the paper out in the next month or so. So I can happily share a link when that's out. So in this next paper, we took a slightly different approach, and we tried to look at a single building typology. So specifically, we looked at domestic violence refuges in Australia. And the reason we did this is because emotional trauma actually can significantly alter brain structures. It can lead to heightened sensory sensitivity through dysregulation of the hypothalamic pituitary adrenal, the HPA axis, which is a primary mediator of our body's stress response system. So individuals with post-traumatic stress, or actually complex post-traumatic stress disorders, often show altered cortisol levels and structural brain changes that can actually affect sensory processing and emotional regulation. And it can create a reduced capacity to regulate responses to sensory inputs. So just to kind of quickly overview here, the stress response in PTSD and CPTSD may actually increase sensitivity in the primary visual cortex through a few key mechanisms. So the first is this heightened arousal elevates neurotransmitters like norepinephrine, which can enhance visual processing. We also see dysregulated cortisol levels, which influence neuronal excitability. And we also see, amygdala hyperactivity, which can increase signal to the primary visual cortex and enhance this kind of threat-related visual processing response. So really what this does is it creates an over-responsive visual system, and it can contribute to hypervigilance and visual discomfort. So that's one of the reasons why we picked this specific architectural typology. So we analyzed 2 purpose-built refuges as trauma-informed design sort of test beds, really. So we took 60 interior photographs across both facilities, and we quantified them using the VISTA tool to measure the visual stress characteristics. And our goal here really was to try and map visual stressors within these environments that may heighten hyperarousal in family and domestic violence residents. So given, what I mentioned, that trauma-exposed individuals may have this heightened sensory sensitivity and kind of over-responsive visual system, identifying and quantifying these visual stressors is really crucial for creating truly supportive healing environments. Oh, this is what one of the outputs from the VISTA tool looks like, though these images are actually anonymized for privacy and security reasons. But from the analysis, what we're able to do is actually identify architectural and design features that may be eliciting these visual stress responses and actually categorize them, kind of categorizing the identified features according to form and also visual material properties in sort of a broader context. So far we categorized 8 features, including acoustic panels, ventilation systems, and LED light flickering, amongst other things. And the methodology, we're really excited about this, and we're actually currently putting together a project to evaluate the inpatient psychiatric facilities in the Harvard hospital system. So we're looking for new ways that we can apply this to specific architectural typologies. And my colleague and co-author, Heather Mitchell, does really incredible work within this kind of space, specific to family and domestic violence settings, that I would really encourage everyone to take a look at. So in the first few papers here, I've really been talking about how architectural environments which deviate from nature may elicit negative impacts on the body. And now I'm going to kind of switch it up a bit. I'm going to talk about another study which we conducted. I think it came out last year, so it's a little bit older, and maybe people have come across it, but it really looks at how biophilia and nature in the built environment can be used to elicit positive impacts within the body. So just to review kind of the theoretical underpinnings of this, the research I've just been speaking about, specifically at the beginning, examined the connection between architectural forms and physiological stress. And similarly, that neuroimmunological research kind of established that connection between physiological stress and neuroinflammation. But to date, no work had really looked at the direct impacts of architectural design on neuroinflammation. And so that's specifically what we tried to do in this paper. So in this study, we wanted to examine the impact of visual exposure to architectural form on neuroinflammatory activity. And so to do this, we drew off of an existing body of research which has looked to establish that exposure to biophilic and biomorphic architectural features can actually reduce physiological stress in the body. And the study really hypothesized a negative correlation between the level of biophilia and the level of neuroinflammatory activity. So in other words, it kind of hypothesized that an increase in biophilic design features would result in a decrease in neuroinflammatory activity. And just to kind of clarify, biophilic design here refers to the direct incorporation of organic material into designs. So for example, incorporating trees and greeneries into facades, for example. Whereas biomorphic form here was used to refer to the integration of naturalistic geometries into design. So for example, the leaf-like structures on Corinthian columns, or the incorporation of specific aspect ratios, which can be found in organic materials. But I kind of use the terms reasonably interchangeably for this project. But biophilic design has been associated with increased frontal lobe alpha wave activity and reduced blood pressure and galvanic skin temperature. So these are all clinical biomarkers which are actually indicative of reductions in stress in the body. And conversely, exposure to urban views, and so by that I mean views of buildings which may host kind of these large volumes of regular, repetitive, rectilinear geometries, is associated with increases in stress. So specifically decreased frontal alpha wave activity, reduced blood pressure, and skin conductance. So as I mentioned, the study really hypothesized a negative correlation between the level of biophilia and the level of neuroinflammatory activity. So this increase in biophilic design features would result in a decrease in neuroinflammatory activity. And so this was a pilot study. It was a very small study. And it was really looking to kind of establish a proof of concept. So in this study, it's actually just 10 participants' neuroinflammatory responses were assessed while they were exposed to 2D images of architectural facades with varying degrees of biophilia for a total of 20 minutes. And biophilic design here was measured, or it was, biophilic design was defined as both the direct incorporation of these organic materials and also the integration of these naturalistic forms and geometries, as I mentioned. But the level of biophilia in each of the respective buildings that I'll show you was measured using a pre-existing framework called the Biophilic Healing Index, which is actually developed by Nico Salangeros at UT Austin. And what the BHI allows you to do really is to evaluate the degree of biophilic features in a building and assign an empirical value to represent this level. But going forwards, I'm actually doing some work at MIT right now to develop a computational approach for measuring the degree and volume of biomorphic and biophilic form integrations. And so should be coming out sometime in the fall, but we're just working through it right now. And neuroinflammation here was actually measured by examining specific types of changes in electrical activity within the brain using something called an electroencephalogram, an EEG device. And these changes in electrical activity were then used as a proxy variable for a neuroinflammatory response. So in the study, the participants' neuroinflammatory responses were assessed by measuring something called the relative power frequency bands using a 32-channel EEG, which means 32 electrodes on the brain. And we were particularly interested in changes across the relative delta band and the relative alpha band, which are specific types of electrical activity within the brain. And these specific types of electrical activity are actually indicative, potentially, of neuroinflammatory responses. And specifically, the changes in these frequency bands can actually indicate changes in something called microglial cells, which are the brain's resident immune cell. And these cells can actually trigger inflammatory cascades of cytokines. So specifically, we're looking at interleukin-1, interleukin-6, and also tumor necrosis factor alpha. And of interest, the role of microglial cells in immune function is actually it's a relatively new discovery. And so, as I mentioned before, for most of kind of modern medical history, the brain was actually thought to be something called immune privileged. So we actually thought that the brain and the nervous system was actually entirely separate from the immune system, which means that those stress responses wouldn't impact neurological activity or cellular function. We now know that's not the case at all. Microglial cells are highly impacted by stress responses, and the nervous system and the immune system are very, very intimately connected. So there's still a lot that we're learning about neuroimmunology. But in this instance, as I mentioned, we basically used the electrical activity that we could measure as a proxy metric for inflammatory responses in the brain, because it's actually very difficult to measure inflammatory responses in the brain. So 2 buildings were evaluated in the study. The first is called the de Kroon building, and it had a low degree of biophilic design features, according to the Biophilic Healing Index. And the de Kroon, it's a mixed-use building in The Hague. It was designed by Rapp and Rapp Architects. It was constructed in 2011. And conversely, the Hyperion City building, which is actually developed by Wave One real estate developers in Berlin and was designed by Studio Pret in Austria, was perceived to have a high degree of biophilic features, according to this Biophilic Healing Index. And the Hyperion City is actually a proposed mixed-use building that incorporates urban farming and greenery and biomorphic and biophilic forms in an attempt to really support resident health. And so initially we were actually looking to use VR, but we've been trying to kind of explore the impact of VR and how it maps to the primary visual cortex. So instead, in this instance, we used 6 images, 3 from the de Kroon and 3 from the Hyperion, and we exposed the participants to each image for 180 seconds for a total of 18 minutes. And the images were really selected so that there was a high degree of similitude, as best as we could, between the de Kroon and the Hyperion building. And so the study really produced 2 primary findings, and these are slightly technical, but I'm happy to discuss in more detail later. But the first was that we saw a decrease in relative delta band activity when looking at the Hyperion City building. And so this connection between biophilia and this relative delta power, I think it's probably the most significant finding from this study, given the association with delta power and neuroinflammation. So this finding actually supported the study's hypothesis. And the second was that we noted no change across the relative alpha band. So this result was actually surprising, given the association between biophilic design and relative alpha power, and also relative alpha power and neuroinflammatory responses. But it may be the result of the protocol, the EEG protocol that we used, but it certainly needs to be investigated further going forwards. But generally, the findings support the hypothesis and suggest that EEG and clinical biosensors can kind of not only be effectively used to evaluate the impact of built environment on neuroimmunological activity, but they also indicate, albeit based on very, very preliminary data, that the built environment may exert a significantly profound influence on human neuroimmunological activity, though this is an area that is sort of ripe for expansion, and we're really hoping that this is just the first step in a number of studies which really trying to kind of get into the details as to why this might be the case and the ways in which the built environment can elicit these responses. But as always, in proper academic form, it's really important that I note that the study faced a number of theoretical and methodological limitations. So this is including, but not limited to, we used 2D unisensory stimuli. Architecture is a multisensory experience. It's experienced in 3D. We also have to kind of talk about the sampling and the data collection techniques used, so specifically the ways in which we process EEG data. The method of statistical analysis, and perhaps most profoundly, is the sample size. We need larger studies. So this is something that we're actively looking to expand upon and engage with. And a lot of the work, if not all of the work that I've shown you today, are really what I'm hoping foundational studies that invite further inquiry. I'm hoping people come and prove us wrong and expand on this knowledge. And it's just the start of kind of a new field that's really developing around this sort of intimate relationship between the built environment and neuroimmunological activity. And I'm really hoping that these research methodologies will become more refined and more nuanced as we move forwards and as the field develops. A key outcome, and actually perhaps the most significant outcome, in my personal opinion, from all of this research has been the growing evidence that the built environment significantly impacts human health, potentially more profoundly than previously understood. And from my opinion, this realization arguably compels us to critically reflect on our role as sort of stewards of the built environment, people who work in the built environment, particularly regarding our ethical and our bioethical responsibilities. So recently, we've been attempting to kind of engage with these questions more directly, and here are some QR codes. Should you be interested, we had, the paper on the right is our bioethics paper that my colleague Heather Mitchell-Tree and I published, which rather is unfortunately behind paywall, ironically. So if anyone would like access, please do email me. I'm happy to send that over. But also on the left is a short piece that I had published in the British Medical Journal a few months ago, which really speaks about the built environment and human health and the ways in which we can engage in these questions more actively. But in our first paper on bioethics, my co-author Heather Mitchell-Tree and I, we really tried to kind of unpack these questions. So, architects, built environment professionals, they're very often under an ethical duty to do no harm, similar to medical professionals. And this is very much a fundamental principle in the professional codes of conduct. So, for example, in the UK, we have the RIBA Code of Conduct, but we also looked at the governing codes of conduct in Australia and in Canada and different jurisdictions. And in architecture, this largely means prioritizing safety, health, and well-being in design, trying to minimize risks, promote positive health outcomes. But of course, constituting a harm can be very complex, and it can involve, very intricate moral, ethical, and legal considerations. And built environment professionals often face challenging decisions, around balancing the needs of various stakeholders. So, for example, I always like to use the example of a public housing project. What's more harmful, not providing enough housing or building in ways that may negatively impact occupants' health? So they're just difficult questions without straightforward answers. I certainly don't have the answers to any of them, but we try to engage with the kind of topic more generally and try and start sort of a discourse and dialogue on this. But in the paper, we kind of argue very, very broadly that there is a crucial need to integrate neuroscience into architectural education, into practice, into policy, so that built environment professionals can really make informed decisions that benefit occupants. But we noted a few kind of key challenges as things stand now. So the first are integration challenges. So there's a significant divide right now that exists between scientific research and architectural practice, which makes it very, very difficult to incorporate neuroarchitectural insights effectively. Second thing is, competing priorities, stakeholder interests, budget constraints, time pressures, they can all conflict. And the last thing really are kind of regulatory limitations. So currently, regulatory guidelines are arguably, in my opinion, not sufficient to actually mitigate the potential harmful effects of the built environment. And this is something that we need to kind of actively engage with. But we also feel that, neuroarchitecture has the potential to really transform architectural thinking, by emphasizing health-centric design approaches. So by focusing on kind of these neurophysiological impacts of spaces, we can actually really redefine standards and practices for the better. But, as we kind of reflect in the paper, there's a few things that we need in order to try and do this. So the first is actually collaboration, so advocating for partnerships between researchers, architects, developers, policymakers, technical professionals to integrate findings into regulations and guidelines. The second thing is actually architectural education, so promoting neuroarchitectural education and kind of physiology education in the curriculum and professional development programs. And the ways in which we go about this are complex, and it's something that we're kind of actively trying to engage with right now. As I mentioned, most of these questions I have no answers to, but we're hoping to kind of engage with people who might know and try and kind of develop these further. And the last is actually the development of accessible tools. So developing practical resources that can actually help practitioners apply neuroarchitectural principles directly into the design process. And this is something we're actively engaging with right now and really trying to explore in more detail, so that it's not the case that we need to run experimental studies for every project. There are potentially ways that we can integrate this into the design workflow, and that's something that we're looking at trying to do. But just to kind of wrap up, what I suppose all of this work is really trying to outline is how the built environment, it may offer a unique opportunity for public health intervention, practically as the impacts of design, are experienced passively, unlike other kind of public health schemes. Our environment affects us, whether we're, when we're just simply existing within it. And arguably there's kind of no neutral space here. It's always impacting us either positively or negatively. So, understanding the impact of sort of architectural form and the built environment on stress responses and sort of neuroimmunological activity more generally is, at least in my semi-biased opinion, both a very exciting research endeavor. The bias there is very clear, but also an ethical imperative. I think it's something that we really need to critically engage in. So we're really hoping that this research continues and that the field starts to develop, and we can kind of explore some of these questions with a little bit more nuance and detail going forwards. So thank you very much.
All right. Thank you, Cleo, for a very comprehensive presentation. Does anyone in the audience have any questions for Cleo?
There's a lot of papers really fast, and I talk really fast too. Hopefully maybe you can slow down on a 0.5 speed.
It was super
quick
clear.
question.
Yeah, of course.
Sorry, go ahead.
I was wondering if your research or anybody that you know have looked at how these types of stimuli might be responded to over like repeated exposure. So if we think about how we use buildings, it's not often that we just go to a building once and never see it again. We kind of go back and back and back, or we live in the buildings. And I think the way the brain works is oftentimes that once it understands that there's some kind of stimuli there, it can dial down its response to that stimuli. So would there be a reduced impact of these types of patterns or motifs if you were to go back to that place time and time again?
It's such a good question. So I actually did a fellowship last year at Harvard Public Health, and the main goal there was trying to understand kind of extended exposure and trying to engage kind of epidemiological frameworks for extended and longitudinal public health studies to try and really engage in these questions. When we're looking at things like those regular repetitive forms on the primary visual cortex, what we're seeing is that the response is reasonably similar with each exposure. But the adaptive response, where you actually might avert your gaze or look away or avoid the environment, is different. We've got one paper right now where we're looking at lecture theaters, which are a particularly interesting case study because you're forced to look at one visual point for an extended period of time. And so we're kind of trying to engage in these long-term research questions. They're very difficult to do because it's really difficult to make someone stay there for an extended period of time. But we're trying to kind of engage with some sort of longitudinal public health methodologies to explore these questions in more detail. There is a question of reduced stress response and adaptive responses within the brain, but it depends on what you're looking at. So with these visual contrasts, it actually seems to be a metabolic response, so that's a little bit different, but it would be more an aversion response. But I do actually think that people kind of recalibrate in some instances. The question is, to what degree, and is it enough to elicit? So when we talk about allostatic overload, it's a cumulative burden. It's all of the things. So it may be the case that, the stress response reduces, but it primes you for additional allostatic responses as a result of like psychosocial stress. So the ways in which all of these responses and environmental features kind of come together, the multisensory element, the psychosocial element, all of these questions will mediate the response that we have. And that's what makes it so difficult. So we're working in like highly controlled environments to try and sort of establish a baseline response. But I think when it comes to real lived experiences, there's so much work that has to be done in order to really understand what that means. But with the visual stress, specifically those contrast patterns, what we actually see is an adaptive response. You just look away, which is interesting in and of itself as well, or avoidance patterns. Does that kind of answer your question?
Yeah, thank you.
The answer is more research is needed, which is so unsatisfying, but it is honest. So
Thanks.
yeah, no pleasure.
Anyone else?
I have two questions actually. So, Cleo, thanks for a very interesting like talk and the papers that you referred to. I have two questions. First of all, you mentioned that right now the regulatory framework is a little bit like not at a very mature stage. So, with all the research that you are doing, are you planning to lobby yourself proactively in order to incorporate some of these components into future regulations? And the second question that I have for you is like from a data science perspective, I know that like you didn't have that much time to go into depth of it, but some of the data that you collected and some of the data science methods that you used, would it be possible for you to share like a paper of what was the architecture of the data science for Penning?
Of course. On the first question, absolutely. And some of the work that we're doing at RISE right now is really trying to engage with EU policy. I mean, it's one of these things where it's an emerging field. And so I do feel as though there's additional research that's needed in order to really establish a kind of solid foundation that we can then make recommendations around. But I do think it's something that we need to actively engage with, particularly around building regulations. I think that there's things that we can do. Visual stress would be kind of a natural choice for that. And also light flicker, expanding some of the lighting regulations. And I know my colleagues, Ian and Arnold Wilkins and Ian Hosking, do a lot of work around that. But I'm thinking and hoping that we can kind of expand that further. And in terms of the kind of technicalities behind a lot of the research methodologies, specifically the computational approaches, I'm very happy to share the paper that kind of sets it out, because we've got our most recent one, which is on the generative AI and Vista analysis, which sets it out in a little bit more detail. So I'm happy to share that in link, or there's the QR code on the video, but I'm happy to send it. If you want to send me an email, I can also send it over.
Sounds good.
I also have two questions, if I could.
All right. Yeah, no, of course, it's a pleasure.
Thank you so much for the presentation. I'm gonna kind of like piggyback what Elliot said regarding the kind of like getting used to or the exposure that you have. Have you also considered as one of the measures like eye tracking and like eye scan path entropy? Because I think also related to it, it was great to hear like in an academic context, like the limitations that we have when it comes to neuroscience, because without acknowledging it, it just stays naive almost. But with all the limitations, for example, for photographs or renderings, and the kind of like the low internal validity of in situ kind of experiments, have you kind of experimented with Gaussian splats or any kind of 3D renderings that is like in virtual reality, where you are pseudo in situ and to kind of like maybe control these external effects that can occur in real world situations?
Totally. That's very much like a watch this space, because you're kind of touching on all the things that we've got going on right now, which is very exciting. I actually think AR and mixed reality provide a tremendous amount of opportunity for neuroarchitectural research, where we get a higher degree of visual control. I mean, again, I work on vision primarily. We've not really moved into multisensory integration, though it needs to be done. It's, that absolutely needs to be done. But mixed reality is particularly compelling, I think, in the sense that we can manipulate spatial characteristics from a visual standpoint while still experiencing things in kind of multi-sensory environment. So it's something that we need to do. I mean, it's one of those things. And I think eye tracking is really interesting as well, because I think that there's a lot of really incredible work that's been done on it, but there's also a lot of questions around what does it mean to avert your gaze? What does it mean if you're drawn to something? I'm drawn to things that I don't find particularly, I'm drawn to all kinds of things, specific environments that I don't necessarily find pleasing, those that I'm kind of intrigued by, that I'm struggling to cognitively engage or comprehend. So I like to look at kind of mixed methods when it comes to clinical biosensors and try and understand what that might mean. I mean, we also spend a lot of time talking about when it's appropriate to use clinical biosensors at all. Sometimes they're not necessary. and actually really being very critical about what the research question is and how best to engage with that topic, and not just use neurotech. I call it like neurowashing, not just use neurotech because it's cool and sexy, but actually because it's telling us something very, very specific. So like with the EEG data, there's a lot about EEG data that we don't understand. We say, relative delta and relative alpha may be telling us something, and that may be quite compelling, but it needs to be replicated, it needs to be expanded, and it needs to be kind of cross-validated in different contexts, which probably comes back to the policy questions as well, whereby, I'll be the first person to say there's a lot more work that's needed to be done, and I'll be the first person to point out all the flaws in our papers, because there's a number of them. There are tremendous amounts of limitations in this space, and I think it's really important that we don't overpromise what our findings are telling us. But what I do think they're telling us is that there's something that's very much worth investigating here, and we need to try and find ways to kind of really engage with these questions in a nuanced way. But it's like exciting, and I think a lot of the emerging tech that's come out in the last 5 years has really allowed us to explore these questions in a way that we've really not been able to beforehand. And the work y'all are doing very much feeds into all of that. So it is very much a pleasure to be here with you guys. Does that answer your question? I kind of went off on one as
I've
well.
imagined a better answer, honestly,
Okay.
both as a scientist as well as a kind of like an aspiring level. Thank you. Genuinely appreciate it.
Okay, thanks.
Okay, anyone else? Seba.
Yeah, I mean, mine is not like question, it's more like a comment. I really enjoyed your presentation, and I think brings interesting elements to discussion, especially when we talk about design. And there are a lot of like rules or concept in design in the design practice that based on your work, it's like you start to question, like the use of certain patterns, geometry. They are all design presets, and like with your work, I know you said that there's a lot of work that it's need to be done, but it's like we are going to some sort of like design informed. I mean, sorry, science informed design. Like less on like some old rules and more into how actually our body reacts to the things it says. So yeah, I really enjoyed it. So thank you.
Thank you. I really appreciate it.
Okay, I guess we are technically at time. Do people have to drop off? Cleo, do you have anywhere to go, or
No, we're all
in
good.
case there's any last minute questions from anyone?
Of course, and also you're very welcome to email me. And if you can't access any of the papers, if any of them end up sneakily behind paywalls, please do feel free to drop me a note. I'm happy to share them.
Great.
Yeah.
I actually have one question if I can sneak it in here.
Yeah,
So it seems like, especially with, for example, some of the EEG work, that there's at least an aspect of it that, I mean, you mentioned that this is sort of theoretically based, a lot of it, right? And so with EEG and focusing on specific frequency bands, the assumption is, for example, that the two bands that you were focusing on, you were looking at it from within the context of sort of more the relaxation aspect. But a lot of the bands have been associated with multiple cognitive functions. And there's also sort of that aspect of context. So resting versus task related states. So that's sort of one aspect. But then the other aspect, it seems as though you said that this is largely positioned within the context of the allostasis model. And of course, there are multiple stress models. And I wonder, I guess, how much the current interpretation now is dictated by that combination of the interpretation of, for example, the EEG frequency bands that you've been focusing on, but also within the context of that specific allostasis model, which is it tends to be more energy expenditure based. And from my understanding, it tends to take more of a reactive stance as opposed to an anticipatory stance. So I guess I wonder if you've thought about how the interpretations might play out if you put them within the different contexts of, some different stress models and also the potential for multiple cognitive functions associated with the different bands.
Totally. So we've given that a fair bit of thought. So I'll kind of preface it by saying that the EEG analysis that we used in this specific instance was very much an experimental model that wasn't really looking at higher order cognitive function. It was specifically trying to identify microglial cell activation, quite simply. So we used existing data that cross-correlated inflammatory blood markers with specific electrical patterns in specific regions within the brain. And that's really what we are trying to focus on. When it comes to kind of like higher order cognitive function in EEG, kind of outside my remit. But in this specific, it's complicated and it's messy. And I think that there's a lot of emerging knowledge that's kind of coming out that's really calling into question things like, relative alpha results in X. I think it's quite complicated and very nuanced. But in this specific case, what we really wanted to look at is we wanted to try and find a non-invasive proxy metric for neuroinflammatory responses. And this is kind of the way that we went about doing that. Certainly needs to be validated. It's not a hill I would die on, but I do think it was an interesting first stab at the question. The second question that you asked is also incredibly related, because what we were kind of assuming was that people are in an inherent state of allostatic stress. You already have a level of allostatic load, and that can be either increased or decreased. So you don't rest at homeostasis. Homeostasis is dynamic, but that a spatial experience can mediate and modulate that in either way. So we had to assume, so we were looking at the difference in the response as opposed to some sort of kind of profound baseline response. So it was very much the delta that we were looking at in the pre-post response of specific delta and alpha wave activity in its relation to microglial cell activation. So it was a very, very narrow research question, which from our perspective kind of helped us to control the scope creep on what that might mean, but it also stayed away from some of the slightly more nuanced complexities of perception, higher order cognitive association with the spatial experience, why you might be experiencing, I don't like bugs. Is it the case that there were some bug, like, I don't, that was one thing that kind of came up, like the biophobia as opposed to biophilia. These are all questions that are inherently related. But what we wanted to look at was whether the geometric composition impacted your kind of ability to visually process the scene, which then resulted in some sort of change in your systemic stress response, which would then be, mediating a microglial cell activation change, which is a proxy for neuroinflammatory activity. So the kind of theoretical causal chain is long and nuanced, but we wanted to try and engage with this question of neuroimmunological responses with specific reference to the built environment. And this was kind of the way that we could do it. In part because we're based in an architecture department. I've got a computational neuroscience affiliation, but we can't do blood samples. Even doing salivary cortisol is quite difficult. We're incredibly limited, but we can do non-invasive EEG. So our question was, can we actually use non-invasive methods to get at really nuanced neurobiological mechanisms? And this was sort of the first attempt. Does that answer your question?
Yeah, I understand that.
Totally. There's
a lot, and I totally get it. So
no, but they're good questions because we've been exploring different models of allostasis, and the fact that there's this idea that we're the homeostatic model is contentious. Some people believe that we exist in a state of allostasis, and then we look for signs of safety that allow us to return to homeostasis or move into homeostasis. Like that is just our perception of one model and how this might act, or how these responses might be elicited. I'm not totally convinced. I think that the allostatic load model is interesting and worth exploring, but I think kind of a, inverted homeostatic model is also something we need to look at. There's a lot of really compelling evidence, particularly from kind of childhood models and baby models. So I think it's something that we almost need to rerun these studies with different theoretical framings, with larger sample sizes and kind of a multitude of biomarkers in order to really kind of get into them. But I also just don't know that we're ever going to totally know. I think that we're going to get closer and closer to answers. and that these insights will sort of change over time. I always kind of die on the hill that like good science is being proven wrong eventually. You know, that that means you're doing it right. You know, so
yeah. No, I completely get it. I just figured that there might
Good point.
be like a starting point to take the results that you have and maybe start thinking about them within the context of some of the other stress models to see whether, you know, might it actually help inform some other potential directions.
I think it would, and I think it would highlight a lot of the gaps in our knowledge right now.
Yeah.
So that's like a public call out to anyone who wants to redo my paper and tell me why I'm wrong. I'm delighted. I'm here for it. I'm here for it. My email's in the end. Come and tell me why I'm wrong. I love it.
Excellent.
Right
All
on.
right.
Thank you so
Well,
much.
thank you so much, Cleo. It was a tremendous talk, very fascinating.
Oh,
And I'm sure there will be some follow-up from at least a couple of people. I probably will personally. And yeah, thank you very much once again.
Hi. Thank you all so much. Lovely to meet you, and thanks for today. I appreciate it.
All right.
Thank you so much,
Bye.
Cleo. Dagmar and I will see you in San Diego. So
Oh, yay. Okay, I'll see you next month. Really looking
Yes.
forward to it. Take care.
All right. Take care, everyone.
Thanks. Bye.
Bye.
Bye.